All right, genetic engineering. This is lecture, I believe it's number 9. In this lecture, we're really going to apply the theories and techniques that we talked about in the other lecture. So this is really sort of the application of DNA replication, transcription, and translation to new ideas and how we can utilize those processes. So a great example here of genetic engineering is what you're seeing in this picture. This is golden rice. The background is that many children all over the world go blind every year because of a lack of vitamin A. Vitamin A deficiency is a primary, I think it's the #1 cause of blindness. About 300,000 cases of blindness every year around the world. So what they did is they put the gene for vitamin A into rice. And rice is a staple for many different cultures' diets, and so by doing that, then people can grow rice that has extra vitamin A, and it's a way of counteracting this deficiency. That's a great example of genetic engineering, adding in a gene to an organism's genome. So to define here, genetic engineering is the direct, deliberate modification of an organism's genome, purposefully altering its genome somehow, either adding in or taking away genes. It can also be called bioengineering. A similarly related field would be biotechnology. Biotechnology is when we use an organism's biochemical and metabolic pathways for industrial production. For example, diabetics have to take insulin. And we used to get insulin from pigs, and diabetics would be given a pig version of the insulin. But what was happening is that some patients would experience horrific allergic reactions, in some cases deadly allergic reactions, because it wasn't a human protein, it was a pig protein. So what they did is they took the gene for human insulin and they put it into yeast cells. Right, and then the yeast cells are now producing a human protein, human insulin, and they can grow the yeast cells in great big vats, and the yeast cells are producing the insulin protein, and then they just isolate out the protein, bottle it, and use it for patients. That's an example of biotechnology, using an organism's pathways to produce some substance that you're wanting. All right. So this is sort of an outline of the different topics that we're going to talk about. I'll come back to this slide a couple of times as we move through. Sometimes we'll get into the nitty-gritty of various techniques, and it's nice to be able to go back and see, what the outline is, where we're headed in this lecture. So let's start at the top here with hybridization. Hybridization is a technique that is going to allow identification of an unknown organism by looking at its DNA sequence. Now, all organisms on the planet have slightly different DNA sequences, right? A little bit different. E. coli's DNA is a little bit different from Serratia's DNA, which is a little bit different from Salmonella's DNA. They all have slight differences in their DNA. Well, what we're going to do is identify which organism we have by looking at its DNA sequence. And this uses what we call a DNA probe. A DNA probe is a short piece of DNA with a specific sequence that binds to only one species DNA. And it has some sort of a reporter molecule attached. I'll say it again. A DNA probe is a short piece of DNA with a specific sequence to bind to the DNA of only one species, and it has some sort of a reporter molecule attached. So in this picture, they're showing here's their probe, right? And the probe there is a little short piece of DNA that binds through base pair rules to only one organism's DNA. It's very specific. One probe will bind to E. coli, and a different probe will bind to Serratia, and a different probe will bind to Salmonella. So the probe is a short piece of DNA with a very specific sequence that binds to only one organism's DNA, and there's some kind of a molecule attached to it to allow you to see it. Usually that molecule is a radioactivity tag. So we can tell whether or not the probe is there because we can see the radioactivity. All right, so here's how this process binds. First step, we'll go through an example. Let's say you have a patient comes in and you suspect that patient has tuberculosis. So you take a sample, you take a sputum sample, you put that onto a petri dish and you grow up that plate and you wind up with a whole dish with multiple different colonies. And the question you're asking is, are any of these colonies Mycobacterium tuberculosis? Because that's what I was suspecting in my patient. So you have a plate of unknown bacteria. First thing you do is transfer that bacteria to a piece of filter membrane, a piece of paper basically. So literally what they do is they put this filter on top of the colonies, smoosh it just a little bit, and then peel the paper off, and the bacteria literally stick to that filter paper membrane. Now you have the bacteria on the surface of your filter paper. Next step is to blow the cells open to expose the DNA. Then they add some more solutions that denature the DNA so it's single-stranded. Then they add in a probe, and when you buy a probe, you know which organism you're looking for because that probe will bind to only one species. So you add a probe that binds only to Mycobacterium tuberculosis, and you wash that probe over the surface of your membrane. Just rinse it over. If the probe finds tuberculosis DNA, it's going to attach through base pair rules. If it doesn't find tuberculosis DNA, it's not going to attach. You wash off the excess, and then you do a little whatever it takes to identify the tag, the probe. In this case, you're looking for radioactivity to see if the probe is there or not. So hybridization here, you have your unknown bacteria, transfer it to a filter, then you kill the cells, blow them open to expose their DNA, then you add chemicals to make the DNA single-stranded, add in your probe, rinse off the extra, and look for radioactivity. If the probe binds to the DNA of an unknown organism, you know what it is. If the probe binds to one of these spots, you know that indeed the patient had Mycobacterium tuberculosis. If the probe doesn't bind, you don't know what that bacteria is. All you know is it's not tuberculosis. So you kind of have to have a pretty good guess before you're going to run hybridization. You have to know what you're looking for. And you can purchase probes for all different organisms. You buy them for whatever it is you want to look for. You can order them online, like you can order anything online. One company I was looking up, they are selling probes for E. coli, Salmonella, Campylobacter, Shigella, Clostridium, Rotavirus, Adenovirus, Mycobacterium, Legionella, Mycoplasma, Chlamydia, HSV, Chickenpox, HPV, HAV, HBV, HIV. So lots of different probes, each one looking for only one organism at a time. So hybridization is a technique to identify an organism based on its DNA sequence, looking for complementary base pairing between your probe and the unknown bacteria. All right, next topic here, genetic tools. Genetic tools, this whole section is going to depend on using enzymes for various different functions. We can use enzymes as tools for manipulating the DNA, for dicing and splicing and reversing and making all sorts of different things. And that's going to allow us to analyze the DNA. So some of these tools here, enzymes that are commonly used as tools, some of which we've already seen, for example, helicase. What does helicase do? Helicase unwinds double-stranded DNA. And indeed, helicase will work just as well in a test tube as it does in the cell. So you can use it in a test tube if you want to unwind double-stranded DNA. Now the other possibility is heat. Heat will also denature or unwind double-stranded DNA. That works very effectively. So if you don't have to worry about damage by the heat, in other words, you're working in a test tube, you could just use heat instead of helicase. Ligase. Ligase seals gaps in DNA. So it seals two pieces of DNA together. Right? It's like molecular glue. Polymerases. polymerases read one strand of DNA and synthesize a complementary anti-parallel strand. Right, so polymerases read DNA and synthesize a complementary anti-parallel strand. Now, DNA polymerases read DNA and make complementary DNA. RNA polymerases read DNA and make complementary RNA. Those are all ones we've talked about before. A couple of new ones that we haven't talked about before. Reverse transcriptase. Reverse transcriptase makes a DNA copy from RNA. That's a very unusual property, a very unusual ability. The reason they call it reverse transcriptase, of course, is because it's doing the opposite of transcription. Transcription makes an RNA copy from DNA, but this enzyme, reverse transcriptase, makes a DNA copy from the RNA, so it's really an unusual enzyme. In fact, this enzyme is not found in any cell on our planet. This is an enzyme that's only found in a couple of viruses. Only one small family of viruses carries this enzyme. HIV is one of them, by the way. So it's a very unusual enzyme in biology, but it's very useful in a lab, so that if you have a piece of RNA, you could copy it into DNA using reverse transcriptase. A restriction endonuclease. Restriction endonucleases recognize specific DNA sequences and then cut the DNA at that sequence. And they're extremely specific. There are a whole bunch of different restriction endonucleases, and each one cuts at a very specific DNA sequence. They're sort of like scissors, molecular scissors for DNA. They can cut through double-stranded DNA. So these are enzymes that are useful in the lab. They're useful in a test tube. You can use them to manipulate your DNA or RNA, whatever it is you have. You can use these various enzymes to accomplish different things that you want to in the lab setting. Let's look some more at restriction endonucleases. Restriction endonucleases, again, are sort of like molecular scissors that cut strands of DNA. Couple of things I want you to know about restriction endonucleases. First thing, each restriction endonuclease has a very specific site that it cuts. It recognizes one sequence in the DNA and it cuts the DNA there. So restriction endonucleases are very specific in where they cut. There are lots of different restriction endonucleases. Hundreds and hundreds of them now that have been identified. And each one of them cuts at its own sequence. So this one that they're showing us happens to cut at CTAG. Happens to cut at CTAG. A different restriction endonuclease would cut at a different location. So each one is very specific for where it cuts. Now, that's the first thing I want you to recognize, is that each one is very specific. The other thing is, I want you to notice that it cuts through both strands of the DNA. It does cut both strands, right, of the DNA all at the same time. Now, in your textbook, they make a big deal out of what are called sticky end cutters and blunt end cutters. We don't really need to go into the minutia here. The only difference is sticky end cutters cut at an angle, like you're seeing here, right, where it has this little overhang. That's called a sticky end cutter for a restriction endonuclease. Other ones just cut right through both strands at the same location. They call those blunt cutters. So if we think of restriction endonucleases as molecular scissors, where each pair of scissors only cuts one particular sequence in the DNA, you can cut with those restriction endonucleases. And if you wanted to put two pieces together, what enzyme would you use to glue them back together? Ligase. Ligase would glue those two back together. Now, restriction endonucleases also allow us to do some analysis on the DNA. Imagine you have a sequence here. This is your piece of DNA. And you're going to cut this DNA with 3 different restriction endonucleases. So you cut the DNA with these different restriction endonucleases, And now you have fragments, you have individual pieces of the DNA. And if you had 3 enzymes, or at least 3 cut sites, how many fragments would you have? Right? You would have 4 different fragments. Now, of course, DNA is way too small to be seen with the naked eye, so how can you see what sort of fragments you have? You're going to run it on what's called gel electrophoresis. Gel electrophoresis is a method for separating DNA fragments according to their size. So here's how it works. You take a gel, it's made very similarly to how we make agar. You pour a gel, it's square like this. You then put your samples at the top of the gel, right? You expose it to a charge. If anybody remembers, what kind of a charge does DNA have on it? I'll give you a hint. There are lots of phosphate groups on DNA molecules. So it has a very negative charge. Phosphate groups have a negative charge. So what you do is you take your DNA, put it at the start of this gel. You expose the gel to a charge, positive on this side, negative on this side. Therefore, the DNA wants to move towards the positive electrode. How fast does it move depends on the size of the fragment. Large fragments travel slowly, small fragments travel quickly. So it separates DNA fragments according to their size, and large fragments don't move nearly as quickly as small fragments. We're going to go through an example of this in just a moment. Here's an actual gel. Right? Here you're seeing this is the actual gel where the samples were put on top, exposed to a charge. The DNA will then move towards the positive electrode, and you wind up with individual bands, and those bands can be measured to figure out how large those fragments are. Let's look at an example of how this technique could be used. Imagine you want to check a DNA sequence. So in this example, you have a piece of DNA, right? You have a test tube, you have a piece of DNA inside of that test tube, and you think you know what this DNA is, but you just want to make sure, right? You're checking to see if this is the gene that you think it is. Remember, DNA is too small to actually see even under a microscope, so we have to go through these sorts of processes to check to make sure that indeed it is the piece of DNA that you think it is. So let's say you have this little piece of DNA. You think that it's a certain gene, gene 3, let's say. Here's the sequence for gene 3. It has already been sequenced and published. Well, what you can do is you can cut with some restriction endonucleases. You buy these restriction endonucleases from a catalog, and you know that enzyme 1 cuts at AGCT, which is here, and you know that enzyme 2 cuts at AGGA, which is here. So if you take your DNA and you add in these 2 different restriction endonucleases, you let them cut the DNA, you now know how many fragments you expect to find. How many fragments are you expecting? 3. There should be 3 fragments after it has completely digested. Not only do you know how many fragments you're supposed to have, but you also know the size that each of those fragments should be. So here it is. You know that this first fragment should be only 3 nucleotides long. There should be a second fragment that's 5 nucleotides long, and a third fragment that's 9 nucleotides long. Well, how in the world can you see if you have the right fragments? You're going to run it on gel electrophoresis. So, here's your gel. Right? You're going to put your sample after it was digested onto the top of the gel. You're also going to add in a standard. This is something for you to compare to later when you're looking at sizes. So in one lane you put your sample after it's been digested. On the other side you put the standard mix. You're going to expose your gel to a charge, positive at the bottom, negative at the top. because you want your DNA to be pulled towards the positive. How fast each piece of DNA moves depends on its size. Smaller fragments move faster. Larger fragments move more slowly. So here's your standard mix where you have one nucleotide fragment, two nucleotide fragments, 4, 6, 8, 11, 15. Remember, this was a mix of all of them together, and your electrophoresis separated them out according to size. Now here's the standard that you can buy from a company as a control to tell how big your fragments are. How many fragments are we expecting to see in our lane? 3. And where are we expecting them to be? 1 at the 3, 1 at the 5, and 1 at the 9 nucleotides length. So that's what we would expect, is to see 3 fragments. The bottom one being 3, the middle one 5, and the top one 9. So restriction endonucleases can be used as a way of checking a sequence. If you have a piece of DNA and you're not 100% sure, you just want to double check before you do another process, you can use restriction endonucleases and gel electrophoresis to do that. I'm going to walk you through it one more time. This is an important subject. So let me walk you through it one more time here. All right. You have this piece of DNA. You want to see if this is the gene that you think it is. You think that you've isolated gene number 3. You just want to make sure if that really what it is what it should be. Well, the sequence, luckily, for gene number 3 has already been sequenced, so you can find it out there in the scientific literature. And here's the sequence. In other words, in your test tube should be this particular gene. You just want to check. So what you can do is purchase some restriction endonucleases. Now, I made these restriction endonucleases up, but there are actually hundreds and hundreds of them, so you can order restriction endonucleases with just about every possible cut site. So for this example, we're using 2 restriction endonucleases. The blue one cuts at AGCT, and the orange one cuts at AGGA. So if you add both of those in with your DNA, you should see the first enzyme cutting there, second enzyme cutting here. So therefore, restriction endonucleases cut through both strands. They're cutting through both strands, you're expecting 3 fragments. And the 3 fragments should be 3 nucleotides long, 5 nucleotides long, and 9 nucleotides long. So after you cut, you want to check your sizes and make sure that you really do have 3 fragments. So you're going to run it on gel electrophoresis. You're going to make your gel, put your sample at the top of your gel, have another spot for your control mix of different nucleotide fragments, expose them to a charge. DNA moves towards the positive electrode according to its size. Here's the standard mix separation, and here are your fragments. Question: what would it mean if you saw 2 fragments instead of 1? What if instead of seeing these 3 fragments here, you only saw 2? What would that mean? It would mean that your sequence was wrong. You do not have the gene that you think you have, because restriction endonucleases don't mess up. They always cut at their particular cut site very reliably. So if you've added in those enzymes and you're getting anything other than the 3 bands that you expect and the sizes you expect, something is wrong with the DNA sample that you have. That's why this is such a handy technique, allowing you to check whether or not you have what you think you have in your test tube. All right. By the way, if you would like to review electrophoresis or hear more about electrophoresis, I did post a video on a virtual lab for running an electrophoresis gel, so you can go check that out. It's in the same it's in this week's folder, week 7's folder. I posted that link. It's to a site on University of Utah's Genetics Center. It's really cool. You should check out their site. They have a lot of cool stuff. But I linked directly to the electrophoresis virtual lab if you want to see that. All right. The next topic I want to talk about is called polymerase chain reaction, or PCR for short. And PCR amplifies DNA. Amplifies is just the fancy science term for copies. So polymerase chain reaction copies DNA. And this is important because often you may have a DNA sample, but you don't have enough to analyze. If you want to go through the processes we were just talking about, where you're going to cut some of it and then you're going to run it on a gel and you're going to manipulate it with various enzymes, if you're going to go through all of those steps, you need a considerable amount of DNA to run those different tests. So what often will happen is you just simply don't have enough DNA. Well, polymerase chain reaction is going to fix that for us. It's like a copy machine for DNA. So this is really important in things like forensics cases, where the forensic scientist will take a little bit of maybe skin samples from underneath the victim's fingernails. Right? So he scrapes under the fingernails of the victim, he gets a little bit of skin cells. He can get a little bit of DNA out of that, but not nearly enough to analyze. So what he can do is run a PCR on those samples, create lots and lots and lots of copies of the perpetrator's DNA, and then run those analysis tests. So PCR is basically a great big copy machine, and we're going to see that it goes through a process where it makes one fragment, makes a copy to 2, and then copies to 4, 18, 16, 32, 64, and you actually get millions of copies within about a 3 or 4 hour period using this technique. Alright, so to run a PCR, here we have our PCR reaction tube. Now if we want to copy a piece of DNA, let's think about what sorts of things we're going to need to put into our reaction tube. If you're planning to copy, you already know some of these things, some of the things that need to be there to make new DNA. So here's the list, the required components here. You need your template DNA. Whatever it is you're going to make copies of, you got to have that. You got to have the enzyme that copies DNA, and that's DNA polymerase 3. DNA polymerase 3 has to have primers in order to function, so you have to add primers. And you have to add a nucleotide mix. The nucleotide mix are all the G's and C's and T's and A's that allows the copying to occur. So if we think of PCR as being like a copy machine, DNA polymerase 3 is the actual machine that's doing the copying. Template DNA is whatever you put in the copier that you want copied. The nucleotide mix and the primers would be like paper and toner and all of those things that you have to add in if you want those copies to be made. So these components are going to all go into our PCR reaction tube together. You add them all in. Here's our PCR machine, and then you add your tube to the PCR machine. Now what the PCR machine is going to do is simply change the temperatures in order to copy the DNA. Right? Really cool. It's just going to change temperatures one step at a time in order to copy the DNA. Let me show you how this works. So here's our template inside of our PCR tube. Here's our template. The PCR machine is going to heat up to 94 degrees. 94 degrees is hot enough to denature DNA. So 94 degrees is denaturing. The two strands separate at 94 degrees. Then the PCR machine will switch to 55 degrees. 55 degrees, primers will attach. Then the machine switches to 72 degrees. 72 degrees, DNA polymerase 3 attaches to the primer and copies the rest of the template on both strands at the same time. So, where you started off with only one copy, now you have 2 copies by the end. So what's the machine going to do now? It's going to go back to 94, which is what you're seeing here. Back to 94, which is going to denature. Back to 55, so the primers can attach. Back to 72 for extension. Now you have 4 copies. Then you go back to 94 to denature. Back to 55 for primers to bind. Back to 72 for extension, copying by polymerase 3. Now you have 8 copies. And over and over and over you go. So you put all of your stuff, all of these different bits and pieces into your reaction tube, put the tube into the machine, the PCR machine, and the PCR machine simply changes the temperatures periodically in order to force these different steps to occur. So it's 94 degrees to denature, 55 degrees for primers to attach, 72 degrees for copying by DNA polymerase III. They call that extension. And then back to 94 for denaturing, and back to 55 for primer binding, and then back to 72 for extension, and over and over and over and over that goes. And you can relatively quickly make large quantities of copies from that DNA. Very effective technique. By the way, in addition to the little video that I was telling you about on University of Utah's site for electrophoresis, there's also one for PCR. It's a really cool little video. I linked to that in this folder, this week's folder as well, so check that one out for sure. It's a great example of PCR. Really, it's a very active, interactive website. All right, so again, PCR amplifies DNA. It makes copies of DNA, and it does that simply by changing temperatures. Now, once upon a time, before they had a PCR machine, we used to do this in graduate school. I remember doing this. They used to have a whole bunch of different incubators. There would be one incubator set at 94 degrees, a little water bath, and there would be the next water bath set at 55 degrees, and the next one set at 72 degrees, and they'd just be right next to each other. And you'd put your tube into the 94 degree incubator for usually a minute, and then you would put it into the 55 for 30 seconds, and then you would put it into the 72 for about 2 minutes, and then back to the 94 for a minute, and then to the 55 for 30 seconds, and to the 72 for 2 minutes, and you would do that over and over again for about 4 or 5 hours. That's how it used to work. But then somebody created this wonderful machine where you just pop it in the machine and the machine changes temperatures for you. Very nice, much nicer than the way we used to do it. All right, so PCR is a way to make lots of copies of DNA, and this is a common thing to do with forensic samples, where you get one small piece of a template, okay, not nearly enough to analyze, so you put that template into your reaction tube along with POL3, primers, nucleotide, put all that stuff into your tube, put your tube in the machine changes temperatures periodically until that one copy eventually becomes millions of copies. Then you can take those copies and go do electrophoresis and restriction endonuclease digest and things like that to analyze the data. So kind of a recap of where we are so far. Enzymes can be used as molecular tools. They work just as well in a test tube as they do in a cell. Gel electrophoresis is a technique that separates DNA fragments based on their size. Smaller fragments moving more quickly, so if you want to separate out DNA, you can use gel electrophoresis for that. And polymerase chain reaction amplifies DNA, or makes copies of DNA. Now, here's the next thing, kind of addition, or next layer I want to talk about with PCR. Good primer design allows the isolation and amplification of a particular gene out of the entire genome. In other words, let's imagine you have a great big long piece of DNA, maybe an entire human chromosome, but you only want to copy one area, maybe just one gene. How can you tell the PCR, only copy that area, don't copy the whole thing? How do you set your PCR up to do that? And it all is about primers. Remember, DNA polymerase III will copy from wherever that primer is attached. Let's look at how this works. So using PCR to isolate and amplify a gene from the genome. All right, remember, primers are actually designed in a PCR. It's a little bit different than replication. In replication, there's that enzyme primase that makes the primers. But for a PCR, you actually design them yourself. You can go to various companies' websites and literally type in the sequence that you want your primer to be. GGCT, AA, GTC, whatever you want. Right? You type in the sequence and they'll make it and send it to you. So primers are designed to bind to a specific place in the DNA according to complementary base pair rules. And by the way, usually the primers that we use in PCR are DNA, not RNA. There's no reason that they have to be RNA. So usually in a PCR, they just use DNA primers. So primers are designed to bind to a specific place in the DNA according to complementary base pair rules. Since DNA polymerase III can only start replication where the primer is attached, the primer attachment site controls what DNA is being replicated by the PCR. So wherever those primers are is where DNA polymerase III is going to start. So if you want to copy just one gene out of the entire genome or chromosome or whatever it may be, you can design your primers to bind complementary on each side of your gene of interest, whatever you want to copy, and that will specify the PCR to isolate and amplify just that gene and nothing else. Let's look at an example. All right, let's say that you need to isolate and amplify only one sequence from a piece of DNA. So we'll do a little example. Here's a piece of DNA. Let's say this is from the human genome, part of one chromosome, right? Notice it's complementary base paired. Better be, I did it kind of quickly, but it should be. It's also anti-parallel, right? You can see it's anti-parallel. All right, and you want to make copies of only the DNA that's inside of that red box. In other words, you want to isolate and amplify just what's in that box, nothing else. Okay. So you want to make copies of just the DNA inside of the box. So what are you going to do? How are you going to tell that PCR to copy only what's inside of your red box? Well, remember, the copying is done by DNA pol III, and DNA pol III starts wherever there's a primer. Since you have to design your primers to add them in, if you want to copy what's there, your first primer will have to be this, GCAA. Right? This has to be your first primer because you want it to bind here. Now, how did I know that this primer belonged on this side of the box instead of on that side of the box? Remember that DNA polymerase III synthesizes DNA in the 5' to 3' direction. So he's always going to head towards the 3'. Right? He's always adding on to the 3'. He's going to go this direction. So therefore, you need to put your primer on the right edge, on the right side. Okay? So my first primer would be GCAA, so that DNA polymerase III can copy the top strand. What would my second primer be? My second primer belongs down there, right? Complementary base paired, and it's got to be at this side because DNA polymerase III is going to synthesize 5' to 3'. So if you can design primers to bind on either side of your gene of interest, when you run the PCR, you're going to get millions of copies of just what's between your primers and nothing else. You will isolate and amplify only your gene of interest. Okay. So again, PCR, if we go back a couple of slides here, PCR is all about copying. And at the beginning, you put in your template, your enzyme pol III, you put in primers that you designed and purchased, and your mix of nucleotides. And then the machine changed temperatures for you in order to make lots of copies of those template strands. Well, if you want to copy only one thing, then you need to make sure that the primers you design are on either side of what you want copied, because that's where DNA polymerase is going to do its copying. So in order to isolate and amplify just one gene from the genome, you design your primers to bind on either side, and that will cause DNA polymerase III to only copy what you want. Okay. So by designing primers to bind complementary on each side of the gene of interest, you can specify the PCR to isolate and amplify just that gene and nothing else. So in our little picture here, if you want to isolate and amplify only what's in the red box, you need primers on each side of the red box, right? One on that side and one on that side. And again, how did I know which side the primer belonged on? Because DNA polymerase III always makes DNA 5' to 3'. This would end up being the 3' end. 5' here to 3' on that end. All right. So, the next topic here in bioengineering is called recombinant DNA technology. Recombinant DNA technology is when we put a gene from one organism into another. So you're transferring DNA from one organism to another. And whenever we do recombinant work, we're always have a genetic donor, right? Whoever's delivering the DNA or giving the DNA. You have a host, the cloning host here, who's receiving the DNA, and you have a vector. The vector transfers the DNA from the donor to the host. So vector transfers the DNA from the donor to the host. Now remember, recombinant DNA technology is possible because of the same code is used for all organisms on the planet. Remember in translation and the activity that we did in lab, translation, that code of which codon encodes which amino acid, that code is conserved between all organisms on the planet, which means if you take DNA from a human and put it into a bacteria or a yeast or a frog, you're going to get that human protein made correctly because all organisms use the same code. Right? So it's easy, it's relatively easy to have that made correctly. All right, so the basic steps here, we have our human donor, we have our vector that's transferring the DNA into our cloning host. Right. We're going to talk more about the steps, but I want to talk a little bit more first about the vector, this structure that's going to move DNA from our donor to our host. There are a couple of different kinds of vectors that can be used. The most common vector would be a plasmid. That's what we're going to talk more about. That's definitely the most common type of vector, those small circular pieces of DNA. The other possibility would be a virus. Sometimes we use viruses as vectors instead. They can also transfer DNA from one organism to another. So let's look at an example here of a cloning vector. This is a plasmid that is often used as a cloning vector. Its name is PBR322. Just by its name, you know that it's a plasmid because of the lowercase p at the beginning of its name. That indicates that we're talking about a plasmid. Usually after that, I mean, people can name it however they want after the lowercase p. Whoever created the plasmid gets to name it. Generally by consensus, BR would be this scientist's initials, 322. He obviously has made at least 322 plasmids. That's generally how they're listed. But you can do whatever you want. I have seen some craziness. Scientists can often be nerds, I'm sure you're aware of, and I've seen some crazy named plasmids. There was one plasmid that was called the Enterprise, and its promoter was called Picard. And the same group had another plasmid that they called the P Millennium Falcon, and the promoter was Solo. Right? So you can name it however you want, but when you see lowercase p, you know that we're talking about a plasmid. All right, now this particular one, this is a map of PBR322. They are different, of course, but there are some things that are constant among all vectors, all plasmid vectors. So all vectors must first have an ORI. Right? What's the ORI for? ORI is the origin of replication. ORI is absolutely required, otherwise this plasmid's not going to get copied during replication. So must have an ORI so that the plasmid can be copied. Plasmid also must have some sort of promoter so that your gene that you put on there can be transcribed. The plasmid must also have room for gene insertion. In other words, it can't be too large, and it also should have a whole bunch of different places where you could cut the plasmid and insert your gene of interest. So for example, here's a HEY2 site. This is a restriction endonuclease site where you could cut a hole into the plasmid here where the restriction endonuclease and then glue in your gene of interest. right, with ligase. And if you look around, there's lots of different restriction endonuclease cut sites on this plasmid. So you could cut it in lots of different locations. That's handy for a plasmid vector. So you got to have the ORI for replication, got to have the promoter for transcription. You have to have enough room to put in your gene of interest, as well as having places to put it, those restriction endonuclease sites. You also need to have a reporter gene. A reporter gene is some way to recognize whether or not the plasmid is in your cells. Right? You need some way to recognize that. You need to be able to tell if the plasmid's there. Very common reporter gene would be antibiotic resistance genes. So PBR322 carries tetracycline resistance gene as well as an ampicillin resistance gene. In other words, normally E. coli is killed by ampicillin or tetracycline. But if that E. coli has PBR322 inside of it, it's going to survive in ampicillin and survive in tetracycline. So you need a reporter gene so you can recognize whether or not that plasmid is there. Let me show you why that's so important. So let's say you have your antibiotic resistance gene on the vector, right? And that's going to allow selection. So here we have, starting out, we have our cloning host. Let's use an example here. Let's say we want to move the gene for human insulin into E. coli. Right? So let's say that's what we're doing. We're moving the gene for human insulin into E. coli. So the first step, you need to isolate and amplify your gene of interest. How are you going to isolate and amplify your gene of interest, which is the human insulin gene? PCR. Right? PCR is going to isolate and amplify that gene of interest. How are you going to make sure that you get just the human insulin gene and nothing else? Good primer design. If you design your primers correctly, you're going to copy only the human insulin gene. Right? That should be review. Then you take that human insulin gene. So here we are. The human is our donor. We've isolated and amplified our gene of interest using PCR. And that's the first step. Then you buy a plasmid like PBR322. How are you going to get your gene of interest, the human insulin gene, how are you going to get that into your plasmid? First, you have to cut a hole. To cut, you're going to use a restriction endonuclease. Then you're going to seal them two together, seal your gene into the plasmid, and for that you're going to use ligase. So PCR, restriction endonuclease to cut your plasmid, ligase to seal them together. All right, now we have our plasmid vector with our gene of interest on it. And that's where we are here. So you have into the test tube goes your cloning host. Whoever's going to get the DNA, in this case E. coli. Put your E. coli in the test tube. You add in your vector that has your gene of interest, right? So your vector is PBR322 and it has the human insulin gene on it. Put that inside. You're going to do transformation. Remember, transformation is the uptake of naked DNA, where the bacteria just pull DNA in from their environment. Now remember, transformation is not a very common event. In fact, it has a very low efficiency. It's not a very efficient process. What can we do to increase the efficiency of transformation? What can we do to increase the likelihood of the bacteria taking in that plasmid? One thing you can do is use a chemical treatment, and another thing you can do is electricity, and that's what I'm trying to show you with this little lightning bolt here. This bolt of electricity goes through it, and that's going to encourage the bacteria to be transformed, to take in your plasmid with the gene of interest. So you put your cloning host E. coli in here, you put your vector that has your gene of interest in there, you zap it with some electricity, trying to get transformation. Now, in all actuality, transformation is not a very efficient process, even when you use electricity or chemical treatment. Even then, somewhere around only 1 out of every 1,000 cells or so actually take in the plasmid. It's a very rare event, even under perfect conditions. Which means most of the cells never took in the plasmid, never were transformed. There's only a couple that were transformed. So if you take that solution and you put it onto regular nutrient agar, what you're going to see is some colonies who don't have the plasmid grow and some colonies that do have the plasmid grow. And you can't tell which one is which one. But if you put that sample instead onto selective media that has ampicillin or tetracycline in it, the only ones that can grow are the ones that have your plasmid. Remember, those antibiotics will kill regular E. coli. Only E. coli that have resistance because of the plasmid can survive. So remember, this plasmid has tetracycline resistance and ampicillin resistance. So if the cells took in the plasmid, then they're going to grow. This is why it's so important to have a reporter gene on your vector. You need to be able to recognize whether or not the organism has your plasmid. Otherwise, you'll spend the next 3 years screening each and every one of these cells until you find one that has the plasmid. If you have a reporter gene, you can find it overnight. Right? Very next day, you would see which cells have the plasmid. All right. So if we put this together and put the different techniques together, think about tracing the creation of a clone, a recombinant organism, in this case a recombinant microbe. We'll use the same example. Let's say that we want to move the gene for human insulin into E. coli cells. Right? So who is the genetic donor? A human. Who is the cloning host? E. coli. And let's say for our vector, we're going to use PBR322. You could choose another vector. Again, you can buy them from catalogs that are online, but let's use the one we know. Let's use PBR322. You've purchased it. All right, so the first step. Here's the human cell, your genetic donor. You need to isolate and amplify your gene. You need a lot of copies of it, and you want only the human insulin gene. So what technique are you going to use to isolate and amplify the human insulin gene? PCR, I hope you said. So PCR, to isolate and amplify the human insulin gene, your gene of interest. How are you going to tell the PCR to only copy what you want and nothing else? Primer design. Design your primers carefully so that it only copies one gene. So now you have millions of copies of your gene of interest, the human insulin gene. You purchase a plasmid, we'll say PBR322. How are you going to get your gene onto the plasmid? First off, you're going to cut a hole into the plasmid using a restriction endonuclease. Then you're going to put the cut plasmid and your gene of interest together with an enzyme called ligase. And ligase will seal those two together, and you'll wind up with this. PBR322 in red, holding your gene of interest in blue. Right? So use restriction endonuclease to cut it open and ligase to seal the two together. Now, how are you going to get your PBR322 with the human insulin gene into E. coli? What's that process called? Transformation. How can you increase the likelihood of transformation? Chemical treatment or a zap of electricity. Now, how do you know if that E. coli cell has your plasmid? It should be able to grow on selective media, either the ampicillin or the tetracycline media. Now from that point, I know it seems like we've done the hard part, but really the hard part is going to be done by the bacteria. The bacteria will now start to produce all of the different genes that are on its plasmid. In this case, the one we're interested in is the human insulin gene. So the bacteria is going to go through transcription to make mRNA from that human insulin gene, and it's going to go through translation to produce that protein, human insulin. If we can grow a whole bunch of E. coli, a great big vat of E. coli all together, they'll make lots of human insulin. Then we can isolate out all of that human insulin, purify it, and use it to treat patients. In fact, that's exactly how they're creating insulin today, except for that not using E. coli, they're using a yeast cell, but exactly the same process. Now, the value of this sort of process is that it enables you to produce lots and lots and lots of whatever your protein is relatively cheaply. Once you get the DNA into E. coli, E. coli is very cheap to grow, doesn't require all of the things that like cattle or goats require. Yeast cells are very cheap to grow as well. It's also very, very safe. You don't have to worry about contamination from outside sources if you're growing these organisms in a sterile environment. All right, so using this technique, you can get all sorts of different products for various different usages here. Biochemical products of recombinant DNA technology. DNA technology really enables the large-scale manufacturing of life-saving hormones, enzymes, and vaccines. Different examples here: insulin for diabetes, I've already told you about. Human growth hormone to treat a form of dwarfism. Human growth hormone we used to isolate from cadavers. We used to isolate human growth hormone from cadavers, but as you can imagine, that was problematic. Every now and then we managed to give somebody HIV, HBV, all of those sorts of things.